Well, while everyone's settling in, happy ILC Day. Welcome to see you, everybody, to commemorate the second Global Lobular Breast Cancer Awareness Day. We're really happy to have you. Thanks for taking time out of your busy schedule to join us today. My name is Sikora. I'm an associate professor at the Cancer Center in the Department of Pathology here. We're really excited again to welcome you all for Global Lobular Breast Cancer Awareness Day. Thanks to LBTA for getting this together over the last year or so with last year's first events. This is a map of all the global events that are happening this year, and then just a set with all the US events. But there were at least 25 events worldwide to commemorate today, which is really exciting. Really great. And due to the fact that 50% of these breast cancers are lobular, and that your community detection that we haven't been getting for years. So just a quick agenda. My goal today is to shut up quickly and we can get to some posters and networking and talk science. But what we'll do today is that I'm going to attempt to give a quick state of ILC in 2025, where we're at as a field, hopefully paint a positive picture about where we've come in the last 10, 15 years, in particular since last year. I'll hand it off to Donna and Tracy to talk more about Front Range ILC, where our advocacy in Colorado is on, in particular over the last year. We're going to do one focused science talk today. Dr. Maggie Musick, a postdoc in my lab, is going to talk about her work in the origins of ILC. And then Dr. Davey Rao, another postdoc in the group, is going to introduce our poster sessions and just a networking table where we can chat about science and advocacy. For our online folks, we are going to try our virtual poster tour, and Davey will talk a little bit more about that. And then hopefully as things wrap up, we can head over to the lab and see some cancer cells. And then please feel free to stop along the way with questions, anything I can clarify, and folks in the lab are monitoring the chat. And so I see all our advocates in the front. The rowdy crowd in the back is on the Sikora lab. I want to say too, I didn't say to start. Thanks so much to the Cancer Center for their support of these events. We couldn't do it without you. Huge thanks to the Breast Cancer Care and Research Fund for their support of our advocacy outreach events, as well as a lot of the Breast Cancer Research Fund. And huge thanks to my lab for putting up with me when I don't know what's going on with this event and for actually being the ones in charge. Thanks, Sam. Let's head to the state of ILC in 2025. So, in hopefully about 15 minutes, I'll touch on where we're at with awareness at the national level, which I really think 2024-25 has been a really outstanding year for thinking about ILC at the national level. I will briefly touch on the fact that clinical trials exist for ILC, whereas about a year or two ago they didn't. So this is great news. And then I will give a very brief research update, kind of to lead into Dr. Musick's talk on her research. So firstly, though, the state of ILC is Colorado. We are the state of ILC right now. So this was a proclamation from the governor's office that just came through yesterday. Thanks, Tracy, for pushing of that one. Proclaim October 15th, 2025 is Lobular Breast Cancer Awareness Day in the state of Colorado. So that's fantastic. And I think this year we're up to well over a dozen states nationwide that have all issued proclamations. So again, really outstanding and civic acknowledgement that this is a type of breast cancer, and we don't want to get lost in the rest of breast cancer awareness. Okay, so since we met last year for Lobular Breast Cancer Awareness Day, there was a couple of big changes for awareness at the national level. In December, there was the San Antonio Breast Cancer Symposium, which at that meeting, there were 50 ILC posters and a whole separate poster highlight session, where about a dozen of those got targeted discussion, which was a big change over trying to fight for space at San Antonio in the past. We talked a little bit about this back in April at our meeting, but I encourage you to visit the LBCA's poster archive, which has lay abstracts for all of these posters as well. Since then, in April, we had the American Association for Cancer Research annual meeting, which is the world's biggest cancer meeting. And we actually had an organ-site research session that was dedicated to lobular breast cancer and the unique histology of the disease, where I had the pleasure of speaking along with Steffi Usterreich at Pittsburgh and Christina Curtis at Stanford about advances in ILC research. And again, that was really new to have ILC featured on that international stage. So that was fantastic. One of the big highlights actually that came out of both those meetings was from the Curtis group at Stanford. So this group back in 2012 published what at the time was one of the biggest omic analyses of breast cancers ever. And this was the Medicare data set, which included over 2,000 breast cancers at the genomic and transcriptomic or gene expression level. And what this study did at the time was separate cancers into their genomic type to help better understand biology and risk. And originally, for ER-positive breast cancer, they separated these into two categories, a typical risk and a high risk. And at the time, though, there was really only one sentence on ILC in that paper, and it said one subgroup, which was the typical risk, was enriched for histotypes that typically have good prognosis, including lobular and tubular carcinoma. And that was it. They just stuffed lobular into the low risk box and got on with it. And that was a disservice to the field at the time because it really did stifle some discussion on ILC genomics for a while. But since then, the group has gone back to Medicare and expanded those analyses. And this was really exciting data that was shared in both of these meetings. And what they actually found since was that, so this is looking at ductal versus lobular cancer in that typical risk versus high risk genomic category. And this is how they define the tumors based on genomic architecture and other tumor features. And they looked at the probability of relapse. And I show this not to scare anyone because we already knew it, but they actually showed this in this larger data set, that whether you look at the high risk category and the typical risk categories, the black line for lobular goes up faster, meaning there was a greater probability of relapse for lobular cancer. And that was regardless of whether they were in the typical risk or high risk category. So this new study that was reported earlier this year said that given comparable genomic architectures, lobular histology remains a high risk feature. And there was a lot more discussion on ILC in these studies. So again, not to scare anybody about the risk of relapse, but what this actually highlighted was that lobular is more than just this low risk signature that came up related to other breast cancers.
Is it possible to use the laser pointer on the screen so the people can see?
I didn't even think about that. Yes, I can do that. All right, thank you. Okay. Yeah, so anyway, it was really exciting that this went from nothing about lobular to now we're actually talking about how these genomic archetypes and features don't define ILC sufficiently, that we need to know more to these. San Antonio is coming up again this year in December 2025, and already we do have scheduled a special session on lobular. This is actually a change over prior years too, where it was only an educational session. This year we actually have new data that's going to get presented on lobular. So these are going to be 4 outstanding presentations, I have no doubt. And then these sessions often have advocate panelists that help dissect the data and give a patient reaction to this. And we're excited that Flora Mujanka, the founder of Bionomy Foundation, who also supports our research, is going to be the advocate for that session. So it should be outstanding. And then the posters haven't been announced yet, but there'll be another poster spotlight. So again, the biggest breast cancer meeting in the world has 2 separate sessions on lobular. So we've really come a long way from begging a few years ago to get any separate sessions on INC. So big changes. Another big change at the national level is the American Cancer Society report that just came out last week. So the American Cancer Society has been putting out organ site-specific statistics for years, overall cancer statistics. And they're really kind of a marketable field of data we all reference about our different cancers that we study. And they actually put out a lot of the specific data set just last week. What this highlighted actually was that they highlighted the prevalence of ILC in the US and also that the rates of ILC are increasing more than other breast cancer types. And we actually didn't really even have that data until recently. So it was really outstanding to not only have this dedicated report from the American Cancer Society, but it also highlighted data we didn't really quite understand, that ILC is 15% and that percent is potentially even increasing. But I didn't actually know this until today, that Good Morning America actually did feature a piece on the increasing incidence of lobular breast cancer. So that's outstanding that we're getting that level of national attention for lobular as a distinct subtype. They did discuss a little bit about lobular breast cancer coming from the lobules, which is still under effect. We'll have to work with them on that. But nonetheless, it's still really exciting to get this level of national attention for your disease. There are ILT trials happening right now, which again, as of a few years ago, that we couldn't have said that. Right now, I was looking through LBCA's database. They do a fabulous job of keeping track of these. There are 4 imaging studies open, many of them comparing PET imaging studies and other screening modalities. There were these two that were based on surveillance and biomarkers, I think including a ctDNA test. And then there are two open therapy trials, one on neratinib targeting HER2 and HER2 mutations and activated HER2, and then one repotrectinib targeting ROS1. Those I think are at Vanderbilt and MD Anderson respectively. Neither open here, unfortunately. The other exciting thing is that we've actually closed about 9 trials over the last 2 years, and a lot of them are still reporting results. So not only are we have trials active, but there have trials that have successfully enrolled and are starting to report data. Some of the first that actually did report last year in San Antonio at ACR. So we're actually now kind of have a proof of concept that we can do lobular specific trials, and this is a thing we can do. We don't have to rely on just digging out the types from overall positive breast cancer trials. So this is really outstanding news for the field. And thinking about ILT in the scientific literature, this is just a plot of papers per year since the first mention of ILC as a word in 1968. And we're doing pretty good. We're up close to nearly 200 papers a year that focus on lobular breast cancer and basal lobular carcinoma. And it's continuing to go up, and we're really excited about how we're contributing to that. Now, while that's exciting, if we think about breast cancer overall in the literature, we are unfortunately still less than 1% of the total scientific literature on breast cancer. That is a huge hill to climb, but undoubtedly there's a lot of room we can catch up with, and we can increase research, we can increase participation by research labs, clinical research groups. So we have a long way to go, but we are on the way up, which is the important part right now. Okay, I did want to highlight just a few papers that have come out nationally before I touch on our group. I am going to send out all these slides, so don't worry about scribbling down details right in a second. I see what you pulled up the phone, yeah. The first one I wanted to highlight was a recent study on imaging from this European, I think it was a European consortium, comparing FDG and FES PET imaging. We've had a lot of concerns over the last few years that FDG was potentially not sufficient as a PET tracer for metastatic ILC. The good news is that this trial painted a much rosier picture about the utility of FDG for ILC. I think my initial takeaway has been that FDG PET may be different, but not necessarily worse than FES for ILC. That there may be certain disease categories and features that FDG does pick up better than FES. So we may have better complementary approaches on top of the recent NCCN change to recommend consideration of FES for metastatic ILC. But I was encouraged that this larger data set, much larger than it did reported before, did have some good news about the utility of FDG. These are a couple studies that were focused on liquid biopsies, circulating tumor DNA and circulating tumor cells to track ILC therapy response and recurrence. One of the big issues we do face with ILC is tracking disease response, in particular in the clinical trial setting, because clinical trials depend on measuring unmeasurable disease to actually define tumor response to therapy. But for so many metastatic ILC patients, the disease may or may not be measurable. So they're excluded from trials. It's much harder to deal with surveillance and then clinical trial therapy response. But these studies, I think, are really great. They have literature to highlight that ctDNA and PTCs may actually be a really unique opportunity for lobular cancer. That in that patients have higher loads of ctDNA and CTCs. We don't know that that's a bad thing per se, but it does give us an opportunity to find biomarkers from distant disease or from things like abdominal metastases that we can't really measure. These are a couple studies from Rita Mukhtar at UCSF. She is tireless in these clinical studies, but we do talk about a lot how chemotherapy is less likely to benefit ILC patients, but we have known that there is a subset of patients that do absolutely benefit from chemotherapy, and we just need to better define those. So Rita's been doing some outstanding work to help understand which patients with ILC are most likely to benefit from chemotherapy, what kind of features their tumors have, and then what chemotherapy response actually means for patients with ILC. How do we define residual cancer burden as a marker of tumor response to chemotherapy? So while I think the general idea that fewer patients with ILC are likely to benefit from chemo, there is a subset that we need to identify, and there's a lot of work in that space. And then this last one was something pretty different in the literature that I was pretty interested to see, in that in working with Laura at the Maggie Foundation, they actually did some survey results from patients and their care teams about the burdens that those groups face for a lot of their patients, given that there are so many different considerations. So it kind of walked through all the different specialties that interact with lobular patients through their care journey and the kind of the psychological burden in considering surveillance and follow-up and disease monitoring. So a really fascinating study, a little different than our usual basic research. Certainly not all that was published this year, but just some things I thought would be fun to highlight. And in our lab, we are certainly not slowing down. Over the last year, we've had, call it 3 publications. One, a medical student in the lab, Michael, he put out with me a review on, we went back to literature to understand radiation response in ILC. Basically, there's been a ton of anecdote that overall lobular tumors are highly radiation responsive, but there hadn't been any reasonable kind of summation of that data. So we went back and tried to pick through the literature, and as you might expect for lobular, there was a lot of retrospective analyses, piecemealing data here and there. But we thought that there was a lot of data to support that lobular do respond particularly well to radiation. And despite that, current guidelines don't really reflect that, and that there is an absence of guidelines suggesting that. We also realized that there is no data firmly on response to radiation for metastatic ILC. So a lot of the data was trying to affirm that we need to improve guidelines to recognize that lobular are likely to respond, but that we do need a lot more new data to support those guidelines, in particular for metastatic disease. And we were excited that some of it got picked up by kind of the Science Free Press, so I got the chance to do an interview and a piece on that, and one of them is there. Another one of our studies that we originally reported the very earliest bits on this back at the 2023 Lobular Symposium in Pittsburgh is that we've been working, and I talked about this last year as well, we've been working on trying to identify how PARP inhibitors may be particularly useful for lobular breast cancer. At present, ILC tumors almost never present with biomarkers that would suggest PARP inhibitor response, but our work suggests that these tumors, in fact, do have a previously unappreciated type of DNA repair defect that's different from the usual defect, or so-called BRCA-NETs, linked to BRCA1 and BRCA2 mutations, and we think it does make these tumors sensitive to PARP inhibition. The paper has been under review for a bit, extensive research, and I really thought we'd have an acceptance ahead of the meeting today. But cross your fingers. And we're still pushing ahead with getting it into the clinical trial setting, which I alluded to last year. Still trying. We're not there yet, but we are trying hard. And then I'm not going to spend too much time on this because Dr. Musick is going to present more about her work. But we did put out a preprint earlier this year on the earliest stages of lobular tumorigenesis and understanding how loss of the gene CDH1 or loss of E-cadherin actually can make a normal breast cell become an ILC cell. And I will refrain from stealing any thunder on that. You'll be able to talk about that in just a few minutes. Finally, one of the other things that we're starting to work on with the Cancer Center and our population health steward resource is the idea that ILC diagnoses, because they are technically challenging, especially for lower volume breast centers that may not see as much ILC, but there's actually a pretty serious disparities issue with the diagnosis of lobular. This is a map with all of our Colorado counties with the relative incidence of lobular from our registry data over the last 20 years. With red being more ILC relative to other breast cancers and blue being less. And what I think is pretty obvious to appreciate is that it's northwestern Colorado, in particular Boulder County, that has a high incidence of ILC. And when we went into this, I said, all of my advocates are from Boulder, so there's got to be something going on up there. I don't think that is biology. I don't think it's environmental exposure. What is likely driving this is access to care and access to high volume medical centers. So I don't think there's biology to the idea that lobular is diagnosed less in southeastern Colorado. So right now we're working to better understand that. And kind of to back that up, what we've identified through our population health data, which is uniquely well built out in Colorado, is that right now one of the best predictors of lobular incidence on a county basis is living in an urban area and living in a high income county, which of course is not biology, that is health disparities. So we're trying to go back to address that, to learn about where these diagnoses are happening and if it is in fact human diagnosis volume and certain, whether it's UC healthcare centers, et cetera. And I don't have to tell you all this, but underdiagnoses of lobular breast cancer is going to limit patient information. It's going to limit their access to any tailored considerations or imaging, treatment, and surveillance that we're learning about is important to ILC. So as much as we're trying to learn about ILC, we want to make sure that these patients are accurately identified so they can get access to all the things that we're trying to bring to you. Okay, I haven't taken any questions on high-level stuff, but we're going to get to that also in networking. But in lieu of questions on there and chat, I will hand it. Oh, sorry.
Yeah, in the two plots that you had, you were defining regular, typical versus high ER concentrations. What are those? What are the limits, or how are you defining those two?
Oh, so these are. Yeah. Okay, thank you. The question was about the high and typical and about ER level. So the high and typical, that is confusing the way that labeled that. It's not related to ER levels. They have a clinical risk categorization that they base on features of the tumor genome, basically how much it gets broken and put back together. That was how they define that one. So there is no definition in this clinical cohort for how much ER is in the tumor. But basically they said that, so this is actually related to our PARP inhibitor questions. When breast tumors have things like DNA repair defects and BRCA mutations, their genome gets broken up and put back together a lot. And the genome has scars of those breaks, almost like wounds in the genome. Lobular rarely have those wounds. That's why they sit in that typical risk category, because of the lack of genomic instability per se, which is also why patients are never eligible for PARP inhibitors, because the tumors don't have any of this genomic instability. So what this is saying is that despite looking genomically boring, low risk, and stable, patients with lobular still have an outsized risk of recurrence, but it's not tied to ER levels per se.
Hi, everybody. Welcome. Thanks for being here. So glad we have our official second governor's proclamation for lobular breast cancer awareness day. It did indeed come in at about 4 p.m. yesterday after I irritated the governor's representative several times. It was like, dude, October 15th, it's the date. I submitted this in May. Come on. But luckily, we did get it in time. So thanks to y'all for your patience, and thanks for keeping me motivated to do that. And part of that motivation comes from Front Range ILC. And so this is Donna. I'm Tracy, for those of you that don't know us. And we decided in 2023 to come up with a local advocacy group for women or anyone with lobular breast cancer, largely women, because we found there were quite a few of us, but we didn't really have any specialized patient advocacy groups, programming, research. Many of us found our clinicians were sometimes not on the cutting edge of lobular. And so we thought, well, if we can help each other out and share information and education, and make sure that incredible researchers like Matt and his lab get support, then it would be helpful to all of us. So as you can see, that's pretty much our mission statement: to provide local patient support and networking opportunities for ILC advocacy, and to provide education on ILC to patients and clinicians in this region.
Sure. And thank you, Tracy, for harassing the governor's office. As Tracy said, we formulated this group in 2023 and have been continuing to try to grow it and reach populations that maybe aren't aware of it. And we'll talk a little bit more about that. We've been trying to offer peer support. The main way that we do that is through, I'm going to jump around here a little bit, is through our primary communication, which is a private Facebook group. We don't necessarily love Facebook, but it is actually the kind of the place to be for breast cancer support in general. There's just hundreds of private groups. There's multiple that are dedicated to lobular, but the one that we've created here with the Front Range ILC is really focused on Colorado. We call it the Front Range ILC because that's where the vast majority of the population is, like where we have a lot of the research and cancer centers, but we of course are open to anyone from Colorado. And we actually have people from all over the country that have joined the Facebook group and are learning and contributing. So we're trying to provide that peer support. It's especially important, as some of you know, from when you're first diagnosed, because it's, you feel like you just got hit by a truck and you can talk to your family and your friends and they're very supportive, but nobody really understands it unless they're living with it. So that's where we're focused on peer support. We also are working harder, I think, is maybe the way to put it, in terms of clinical resources. So we leverage a lot of the Lobular Breast Cancer Alliance, LBCA's materials, because they are the premier source of materials and they're a well recognized and respected organization. And so when we're talking with clinicians, whether that be oncologists or social workers or oncology nurses, that helps us to educate them. Of course, research updates, working very closely with Matt's lab for these twice a year gatherings where we learn about the research. We've also been trying to share some of that research regularly on the Facebook group so that people are aware of it. We now have about 100 members writ large. So that would be people that are both on Facebook and people that are not. So anybody who has signed up to participate in one of these outreach events who's not on Facebook is counted as part of the Front Range ILC. And while of course we don't love to see high numbers of people diagnosed with ILC, we want them to find us. And so as you are, talking to people, any of you, and if you happen to hear somebody mention that they had a breast cancer diagnosis or they know somebody with it, just kind of gently ask, like, oh, do you know what type of that is? Because, we would love to welcome them in. We do have a dedicated email that goes to multiple people. So if you want to try to reach somebody and you don't know the email address, you can always email to the Front Range ILC 2023. And the last thing that we kind of wanted to put out here is, we're here to try to guide the group, but we really want to hear from everybody who's a part of Front Range ILC, as well as, and that includes the Sikora lab as a part of ILC. So a couple of months ago, we sent out a survey and asked people to respond. And we were kind of interested in what kind of support people might want. Do you want to meet regularly, like virtually, or have coffee meetups? Are you good with meeting twice a year for these more formal events? And we got a couple of responses, but it wasn't the response rate we were looking for. And so the plan is to resend that survey. If you've already filled it out once, which I know some of you did, you don't have to submit it again. But I would encourage everybody to just give a little bit of feedback. And Matt has kindly agreed to include a link to that in his follow-up from this event. So you can get the survey there or on Facebook. The other thing that we would really love to hear from you, and maybe later today, as we're kind of having some discussions and visiting posters and things, are things that you might like to see the group do. One of the things that we've been talking about is doing a dedicated fundraiser to help support the Sikora lab. I would also encourage all of you, if you are interested and have the means, to go to the Sikora lab website. It's very easy to make a donation of any size, even if you just want to forego that $6 cup of coffee, or something more substantial, and you might even get some good swag out of it. The other thing that isn't listed here, but I want to thank Matt and Davey on behalf of Tracy and myself and Carolyn and Judy, who've joined the leadership team, is that we now actually have a webpage for the Front Range ILC, which is one of the pages that's on Matt's lab. And we felt that that was really a smart way to have a web presence, because that means we didn't have to maintain a separate site. And then if people found us, they found Matt, and vice versa. We've done a couple of outreach events over the past couple of months. Most recently on Sunday, I didn't make it out, but you can see pictures there of all the people, Matt, Judy, Carolyn, and Tracy, and some pups. This was in Denver, the American Cancer Society, and there was a booth there with all kinds of great information and a pumpkin to draw people in. There was a Colorado Cancer Fun Run in El Dorado Springs in September. And then Matt and his lab have been doing kind of these tours, I guess, around the state. And one of them was in Greeley in August. So Carolyn and I went up and met Davey and Matt and talked with a few people that came through for just focused on the local community. And these are run by the Cancer Center at Anschutz, and then advertised locally. And one thing that I really was gratified by is that as a woman had come by the booth, and so I started talking with her, and she said, "Oh, well, I'm on your Facebook group, and I saw that you posted this, so I wanted to come." And so we're reaching people. It may be one at a time, but that's okay. But it's really great that we were able to participate in this, and I'm sure there's other opportunities. And apparently Davey didn't get the memo to wear a printed top, but we'll let you pass this time. Okay, whatever.
Thanks. I think that sums it up. Yeah, we're trying to explore other avenues for groups to get together besides Facebook, but it is the devil we know for now, so it's the devil we deal with. It is in our survey, though. If you have other ideas or thoughts about ways to communicate, whether social media or email, please let us know. Just these photos are kind of evidence of the growth of the group over the last year and our participation and our getting out there. The Ask Me About Lobular breast cancer is very catchy, and we appreciate LBCA donating that to us because people actually do ask about it. And finally, thanks so much again, as always, to the Sikora Lab for giving us a platform to provide support to other ILC patients and to partner with doing research. And if anybody has questions, we'll have a whole table for advocacy, so you can swing by and chat. Anything online?
Oh, I just wanted to mention there's another research roadshow this Friday in Steamboat with the Sikora Lab attending.
The next research roadshow will be this Friday in Steamboat, the Sikora Lab.
Just also mention there's brochures if people want to take them to their clinic.
And any patients or anybody else who has a relationship with a clinical organization who would like to take any literature from LBCA, we have tons of brochures. Please share, spread the word. That's what they're here for. As many eyes as we can get on them, as many people we can make aware. So
I should have done this first, but Carolyn, would you mind standing? I know the people online can't see you. Carolyn, she and Judy, please stand. Both of these ladies like consistently would like message Tracy and I or chat at these events. And it became clear that like, hey, they're invested in this. And so we recruited them and we are now the leadership team. But it's not an exclusive group. Basically, if you just ask a couple of questions, we'll draw you right in. So thank you both because your contributions have been very impactful in this last year or so.
All right. Oh, I'm a loud talker. Okay, so I'm Maggie Musick. I'm a postdoc in Matt's lab. I've been a postdoc in Matt's lab for 3 years now. And today I wanted to share with you the work that we're doing on E-cadherin and the origins of ILC. And I am a big pointer, but I'm going to really try to use this so people online can follow along. So our most recent publication in the Sikora Lab is on E-cadherin loss and the initiation of ILC. And as Matt said, these slides will be shared, so you'll have a link to this paper. So one of the hallmarks of ILC is E-cadherin loss. And we know that E-cadherin loss occurs very early in the progression from normal breast cells to ILC cells. We also know that E-cadherin loss alone isn't sufficient to change a normal healthy breast cell into an ILC cell. needs other changes to occur. But we do know that E-cadherin loss is very important, plays an important role in the progression or the development of ILC. So why do we care about E-cadherin loss? E-cadherin is a protein that's found on the surface of breast cells, and it's important for cell communication and for cell identity. And when two breast cells with E-cadherin communicate with each other, they're able to appropriately regulate movement, growth, and survival within the breast tissue. However, in ILC cells that don't have E-cadherin, they're not able to communicate in this way, leading to irregular patterns of growth and survival. However, we still don't know exactly how E-cadherin loss pushes these normal healthy cells to being more towards a cancerous state. And the majority of ILC research, or rather what makes this question, what has made this question difficult to answer, is that the majority of ILC research has been in ILC cell lines, which have E-cadherin loss, but they also have all of those other changes that had to occur in the breast cells in order to get to the state of ILC, making it very hard to tease apart what exactly the role of E-cadherin loss is in ILC. And so what we've been doing is using human mammary epithelial cells, or HMECs, and these are primary cells derived from various donors, usually from reduction mammoplasties. And so far, we have been using models from 4 different donors, 2 from young donors and 2 from aged donors. And this will eventually let us ask the question of whether the genetic alterations that happen in the aging process play a role, if they're important for this progression of ILC or not. We can contrast that with the young donors. And so what we did in this work so far is we've looked at the primary cells with E-cadherin loss and just asked, how is E-cadherin loss changing these healthy breast cells and pushing them towards an ILC state? And so breast cancer, including ILC, originates in the ducts or lobules of breast tissue. And specifically, there are 2 main types of cells that make up these ducts. You have luminal cells, which line the inside of the duct. And these cells are hormone responsive and express the estrogen receptor. And ILC cells are typically categorized as luminal cells. And then also outside of the luminal cells, you have a layer of basal cells, which are not hormone responsive, and they actually have the ability to contract during lactation to pump the milk out of the breast. And so these are 2 very different cell types, very different functions, different identities. And the nice thing about the models that we're using is that they have both of these cell types, luminal and basal cells. So what we did was we sorted the HMECs into luminal and basal cells, and then we deleted E-cadherin in both luminal and basal cells separately to ask how is this affecting luminal cells? How is it affecting basal cells? Is it the same? Is it different? And interestingly, what we found was that E-cadherin loss in luminal cells and basal cells reprogrammed both of those cell types to the same new identity, new cell type. And this new cell type has a lot of similarities with ILC. Some of the cell functions, some of those cell programs are similar to ILC. And these cells are categorized as luminal cells. And they also have increased growth. So we have a long ways to go from this point to really look at the progression from these healthy breast cells with E-cadherin loss to follow through with further progression along the ILC development. But so far, we've found that E-cadherin loss is reprogramming these cells to the same potentially precancerous state. And so what does this mean for the ILC community? So breast cancer subtypes, IDC and ILC, can be categorized according to their hormone receptor expression and their molecular subtypes. And as you can see, ILC is predominantly estrogen receptor positive, and this molecular subtype called luminal A, which just means that it expresses the estrogen receptor as well as other hormone receptors. And now, because of the work that we've done, we can actually say that E-cadherin loss reprograms luminal and basal cells to the luminal cell identity, which is why ILC is typically classified as a luminal breast cancer. And so where do we want to take this research? Where can this research go? Down the road, our ultimate goals for translational findings are to develop new biomarkers that can help better predict the progression from ALH-LCIS to ILC. For instance, if we could identify E-cadherin loss plus gene X increases the risk of LCIS to develop to ILC. This could help improve treatment confidence for physicians and for patients in deciding whether their treatment path is going to be surveillance or maybe surgery or chemoprevention. And also, we'd like to identify vulnerabilities related to E-cadherin loss using these same models. And that would potentially develop into new treatments that could be ILC specific and potentially prevent this progression from LCIS, benign ILC precursors to ILC. And that is it for the summary of the work that we published. And I also have a poster if you'd like to talk to me later.
So I was wondering, what does it take to watch one of the cell lines or any of the cell lines progress from normal through the loss to ILC?
Good question. Judy asked, how long does it take to watch the cells progress from normal all the way to ILC? We haven't done that yet with these models, but the model that I use the most, that's most similar to ILC, it takes 2 weeks. It takes 2 weeks to see the E-cadherin loss. And we have to have some really neat time course data within those 2 weeks, seeing some changes that are happening. But we have not gotten that far, but that is the plan. Done.
I have two questions, but one I'm going to save to
Okay.
ask you. But maybe the easier one to answer is, could you give like the layperson's definition of luminal A versus luminal B? Because it looked like in that other slide that it's luminal A that is predominant with ILC and not B. And I'm not sure I understand the difference between them.
Yes. I believe luminal B is also HER2. If Matt will help out this one.
That's a good question. So first, before I answer the question, I'd point out that these subtypes, luminal A, B, basal, normal, HER2, are based almost entirely in ductal biology. The original definitions were from a study that included like 100 ductals and 5 lobulars, all of which were weird. So I would argue that this doesn't mean what we need it to mean for ILC. It is still informative for risk. Luminal A in general is ER/PR positive, HER2 negative in general, and has lower proliferation. Luminal B can be HER2 positive. It's kind of a mixed bag. Is generally PR negative and would have a higher proliferative rate to Ki67. So if you can use the 4 histological markers, ER/PR, HER2, Ki67, to loosely define A and B.
Can it switch from A to B?
Yeah, so there can subtypes switch. And yes, so we, over the last few years, have gotten more data on ILC switching. Luminal A to B is probably pretty subtle as far as biology. PR loss can be indicative of a reduced hormone responsiveness. So we've been hand waving for decades in the field that loss of PR may be indicative of a reduced endocrine response and loss of response to anti-estrogen therapy. Tumors can gain HER2 and switch subtypes. Tumors can lose ER altogether and switch subtypes. They vary within ER. So you typically like lose things as opposed to gain things in the progressions. But again, all of this is based on IDC biology, and there's at least within luminal A ILC, there are at least 2, maybe 3 transcriptional subtypes of ILC that we're trying to understand better. So this is only a loosely useful framework for ILC.
So cancer that originates in basal cells, does that happen? Is that a different type of cancer?
That's a good question. There's different, oh, yes, sorry. Are there cancers that originate in basal cells? So that's a good question. That's kind of what we were starting off with E-cadherin loss was we thought maybe since ILC is mostly luminal, that E-cadherin loss in basal cells would just kill the cells and it wouldn't be able to progress to any sort of cancer. But we found that it actually programs basal cells. So we don't have an answer for ILC, whether it can start in basal cells or luminal cells. We don't have a cell of origin yet, but there are other oncogenic changes that have different effects in luminal versus basal cells.
Yeah, I'll go into that. It's a very unfortunate naming system that the group that made this used luminal and basal, because really it means luminal-like and basal-like, and it is loosely tied to the cell types of origin. Hand-wavingly, triple-negative breast cancer would come from a certain subset of luminal cells or basal cells, is the short version of answering your question. So yes, it can, but it's hand-wavy. And we're only just in the last few years getting more human data on these cell types. A lot of the cell type biology is based on mouse models because we have just more data from that setting. So we're better understanding in the last few years about the human subtypes of luminal and basal cells.
It just seems that these cells have different functions when they're healthy.
Yeah.
And that they then both
So
like that.
Yeah, the comment that they have such different function is why Maggie's data that the E-cadherin loss makes them revert to something the same, why we were so excited about that, because we thought that had no business happening whatsoever. But it definitely changes how we're interpreting written paradigms for ILC, and it may not yet, but I think it will give us insight into how parity, breastfeeding, and related things like aging are related to lobular, because it is different. But we don't have a good handle on any biological reason why. But the fact that it can impact basal cells similarly can give us some insight there.
Good, everybody can hear me? Awesome. Hello, everyone. For those of you that don't know me, my name is Davey. I'm a postdoctoral fellow here in the Sikora lab. I joined here in February of this year. And today I wanted to just briefly go over the poster networking sessions that we're going to have immediately following me talking about the poster networking sessions. And in between that, there's going to be a little bit of a break. So for those of you that are here, please, restrooms just around the corner, and we do have snacks in the back. I want to remind everybody, please partake. And then Matt got the really good cookies. So, first up, I wanted to talk about the posters that the Sikora lab will have just outside of this room. And there's a decent amount of them. And I also wanted to just kind of reinforce how much we all really love the work that we're doing. As is evident by the fact that 3 of the people that are presenting posters today don't actually work in the Sikora lab anymore. They're just back here for today's event. And so I'll go through those posters. Very briefly so you can kind of have an idea of who's presenting them and what they're presenting. So if you're interested, you can check those out. And then I'll explain, we're going to have 3 different tables for those who are here in person, where you can chat with a couple of different groups. And we also wanted to make sure that those of you that are online, you are able to participate in either the poster session or these table events. And so I'll explain how we have set up this virtual poster/table chat session as well. But first up, let's talk about posters. So first up, we have Dr. Joseph Sotnick. Joe, would you like wave and so everybody knows who you are? Joe is our lab manager. He's the one that ensures the lab runs smoothly, and he's the first person to go to with questions. So not only does Joe do that, but he also does a stupid amount of research in the lab on ILC, and as is evident by the fact that he has 2 posters that he will be presenting, not just one. So the first poster that Joe has up is going to be talking about the estrogen receptor's control of the invasive lobular carcinoma genome. So a while ago, we identified that estrogen receptor works very closely with a protein called MDC1, specifically in ILC, and it does some unique things with how interacting with estrogen receptor and how it regulates estrogen receptor regulated genes. And so if you want to learn more about what MDC1 is doing with the estrogen receptor and how it's kind of unique in ILC compared to other cell types like IDC or no standard or no special type, I can talk, I promise. Go check out that poster by Joe. His other poster is pretty different, but it's no less exciting. And this poster is focused on developing new models of metastatic ILC. We know that ILC tends to find itself in different specific locations compared to other types of breast cancer, but we don't have fantastic models for researching those unique metastatic sites. And so Joe has been working very diligently on trying to develop new models so that we can better understand these unique sites with using a special technique called MIND. It's a really fascinating technique. He has become a pro at it. So please go talk to him to learn more about these new models that we're developing in the lab. You already heard Maggie's presentation on the loss of E-cadherin and how it relates to the development of ILC. She's got a very beautiful poster in the back where she can go into more detail, dig into the data a little bit more. I won't really say anything as Maggie already said it all, but you should check that one out as well. Up next, we have Maddie. Maddie is one of those people that, sadly for us, no longer works in the lab. Maddie was a technician for 6 years. She's nodding. In the lab, but just recently left us to go on to start her PhD. So she is currently a graduate student here on campus in integrative physiology. So even though she is very, very busy with her first semester of classes in graduate school, she elected to come back here and talk to you about the amazing work that she did while she was a technician with us. I also want to state that this poster was given earlier this year at the STEM Poster Day, which is an event that is held at the Colorado Capitol each year, where researchers from across Colorado come together to talk about their research, not only to the public, but to legislators. So that's an event that should be happening again next spring. But Maddie, as well as another individual from our lab, presented posters there. So these are really excellent posters that talk about the science, but also don't tend to get lost in the nitty gritty that a scientist loves so much. So the 2 topics that Maggie will be talking about with this poster. The first one is looking at how ILC cells tend to do things a little bit differently in terms of DNA damage response. And then the other portion is going to focus on another protein that we are interested in, specific to invasive lobular carcinoma, which is called WEN4. And WEN4 appears to be doing some bizarre things in ILC, and looks like it is playing a role in how the cell is able to get energy. So talk to Maddie about either of those 2 things if you are interested. Changing gears just slightly, we have Dr. Taylor Benson. She is currently another postdoctoral fellow in the Sikora lab. And Taylor will also be talking about WEN4, but instead of focusing on invasive lobular carcinoma, she is instead switching to talking about ovarian cancer. And so, as we've mentioned, the work that Maddie has done focusing on WEN4, I see everybody giggling in the back. Did I say something funny? Okay, good. Trying to make sure. Taylor has been focused on went for. But as opposed to ILC, she's going to be focusing on the role that went for plays in ovarian cancer. So there are actually some variations of went for that in ovarian cancer appear to be changing how the cells are able to access the energy they need to be able to grow. Again, this was another poster that was at the STEM poster day, so it's a really excellent poster to talk to Taylor about, even though it's not ILC. Next, we have Carmen. Carmen was one of those that left us but has come back today. Carmen was a master's student in the lab who did her thesis with us, but has since moved on to industry. But she is back today to talk about the work that she did for her thesis and kind of relating to Joe's work on looking at this unique protein MDC1 and how it relates to the estrogen receptor and regulating estrogen-regulated genes. This is a really cool poster because it, as Matt calls it, is the Lobular Breast Cancer Research Fund in action. So the money that was donated to this fund actually allowed the lab to be able to purchase some really cool specialized equipment and to do some really experiments as well as analysis looking at what exactly MDC1 is doing in ILC cells, what cellular states are we seeing that are within this population of ILC cells. And so check that one out. It's really cool to figure out how this equipment and analysis, which is called PIPSEQ, works. It's pretty fun. Next, we have Evelyn. She was one of the last people that has left the lab that has come back. Evelyn was a summer student this year with us. She's currently a senior at CU Boulder studying engineering, but she elected to spend the summer dipping her toes into biomedical research with us. She is also the daughter of Donna Charlevaux here. And so we were really, really privileged to have Evelyn join us for the summer. And she will be talking about the research that she did looking at how she was evaluating a new class of estrogen, anti-estrogens impacts ILC. So 3 different terms were SERDs, and how they are impacting the expression of the estrogen receptor, and then also how they are impacting gene expression. And then finally, I have a poster, very less sciencey based. I was able to give this poster earlier this year at a science communication conference where I really wanted to highlight how the collaboration between scientists and patient advocates has helped benefit both the scientists, hopefully the patient advocates, but science as well moving it forward. So if you would like to learn more about that, I would love to have you at my poster. Okay, so those are all the posters. They're all going to be in the back outside of the room if you want to chat with any of us. We're also going to have 3 separate tables that will be set up in this room. And the first one is going to be a man or woman, I guess, Donna Tracy. This is going to be focusing on Front Range ILC. They're going to be talking about patient advocacy in general, the events that they've held in the past, as well as hopefully future events, and how you can get involved in this patient advocacy group. Then this table is going to be actually manned by Matt and such. So he's going to be talking about mainly the Lobular Breast Cancer Research Fund, what it is, how it got started, what it means for the lab and for research, but also just in general. Come say hi to Matt. He loves talking about the work that we do and the science that we do. So if you have any questions, please stop by and talk with him. And then finally, the last table, I'm going to spend just a little bit of time talking about this. I will be at this table, and I'm really hoping to start developing a lobular breast cancer focused podcast. So we are currently titling the Lobcast. It's something that Matt and I have been discussing over the last couple of months. The idea here is that we want to create a podcast that is focused on providing information for individuals who are newly diagnosed with ILC or researchers who are stumbling their way into the science behind ILC, and provide information for patients, advocates, researchers, clinicians that currently isn't available in an audio format. So if I would stop by, I would really appreciate that. I'm going to try and set up some official podcasting equipment to hopefully just kind of pick everybody's brain on if they think this would be a good idea. Hopefully, yes. And then if so, what aspects about it they would find most beneficial as we actually work into developing this, because we want this to be a resource predominantly for patients and patient advocates. So I would very much love it if you could stop by that table. All right, so that was all the in-person poster and tables, but we want to make sure that those of you attending online are actually able to access some of these. And so what we have done is we have asked our rotation student, Natalie, in the back to moderate this session where essentially every 10 minutes, we will have either Matt or one of the poster presenters sitting in front of a computer over Zoom, explaining their poster to you over Zoom. So first we'll have Matt, again, just stop in, say hi, chat with him for a little bit. Then Joel will be talking about his metastasis models, Maggie talking more about E-CAD, loss and ILC, then Taylor talking about one form of ovarian cancer, followed by Maggie, who's going to be talking about highlights in ILC biology, and then ending with Carmen talking about her cool seek data. And so at any point, please stop in to talk with those researchers about the science that they're doing. And Natalie will be there to monitor the chat, make sure all of your questions get answered, and to make sure everything goes smoothly. So I think that's what we have for right now. We can set this one at like 12 or 2:45 or so. It was a little under 10 minutes for a break. If anybody has any questions over the chat, please let us know. If you have any questions in person, saying anything.
All right. Oops. It looks like we got the audio fixed. Can everyone that's online hear me? Awesome. Okay, we will have Matt over momentarily. He's on the way. It looks like it's good.
Okay. All right, I think we're good. Any questions? Thanks all for being here today. We really appreciate it.
Thank you. May I ask a question? You didn't mention the trial, the Clever trial at Penn?
Oh, did I miss one? I'm not surprised if I missed one.
Oh, no, but I just wanted to ask. I'm wondering if this will become standard of care, checking for residual tumor cells, residual cancer cells in the bone marrow to basically get ahead of stage 4.
I love those ideas for looking at residual tumor burden. For bone marrow, I don't have a great answer for you for how it's going to change care for ILC specifically. I can tell you that one of the projects we have is trying to understand how lobular cells interact with the bone differently than other breast cancers. We are seeing that similar to how they spread in the abdomen, they do make the bone behave in different ways. It actually changes the relative ability of the bone to produce bone versus break down bone. And I think that will eventually lead to how it colonizes the bone marrow or not. So we're trying to understand at the basic level. I hope that does translate to improved monitoring, whether it's the disseminated tumor cells in the marrow, things like circulating tumor cells or ctDNA. So I'm hopeful that we do learn more about that. It is interesting that lobular does live in those circulating spaces more, but we don't know if that's a bad thing yet, or if it's just a feature of the cells.
Interesting. All right. Thank you very much.
Yeah, thank you for joining us very early for you.
That's okay. It's worth it. Thank you.
Yep, thank you.
Yeah, so there's.
Any other questions?
I guess one more, if I can ask another.
Please.
Christina Curtis's research on the histologies. From the papers that I was able to look at, she goes through 4 distinct histologies, but not the others. And I'm wondering where I can get where I can get a study with the detail of all 9 subtypes.
Are you referring to her integrative subtypes?
Yes.
So in her most recent one, they grouped a lot of them. They're all the data is still there on the 13 subtypes.
Yes.
It's all there. They just group it together differently. I think because they realized that some of them could be grouped without losing clinical power. But if you dig through the supplement, all of the individual definitions are there. They do have a couple tables that actually break it down by either they have a couple different ways they define integrative subtypes, kind of like they made like a simplified version. So there is one table that does break it down on a per tumor basis if you want to dig through the data.
Okay. All right. And they were looking at the mutations. And what struck me was some of the mutations are not in the MSK impact report.
Hmm.
They're not covered. So would somebody just like, would you go to a lab and ask them to look for certain mutations in a tumor sample?
No, it's tough. I mean, MSK had to make some tough decisions about what to actually cover in their sequencing panel because
Right.
For the most part, those panels are not just sequencing every single gene. You're actually restricted to a number of
Right.
mutations.
505. Yeah.
Right. So yeah, so if a study does have the ability to do things like exome sequencing, then they
Yeah.
do get more coverage. And yeah, it's a cost benefit ratio. You know, we're planning right now for doing some of the mutational studies in LCIS that I was alluding to. Like, can we track
Right.
which mutations follow LCIS progression or not? And we're playing the same game of like which panel has the coverage versus what can we pay for. It's an unfortunate game, but I think some of the things that MSK doesn't cover are perhaps uncommon enough that you're not worried about that mutation from a tracking standpoint per se, because you should be covered by other mutations because it's not like it's the only one in the tumor.
Right. Okay. But if you're sitting at 1% rarity of lobular, you're scratching your head wondering which one of these do I fit into. But
oh yeah, no, it's certainly and that's a problem with those data sets in general. And you know, I think we'll get better. A lot of the groups that are going to be presenting at San Antonio this year do have the capacity to do that. Especially the European groups have been like pushing really hard to expand lobular in those data sets. So I think MSK is going to get run past real fast.
okay. If there's one thing that could help you to advance and accelerate your studies, what is it?
Aside from sustained funding. I mean, that's most of it. You know, I have a really passionate team that, you know, I think we all alluded to that a lot of them came back to hang out and talk to everyone today, even though they don't work for me anymore.
Right.
So I'm not hurting for that. For us, it's the cyclical nature of grants is hard to predict sometimes. And right now with federal funding the way it is, it's hard to predict a few years out, and it's hard to get people to join the lab full time. And I'm trying to recruit Natalie here to join the lab full time, you know, for a 5-year graduate program. And, you know, it's hard to know what my funding is going to look like in 5 years. But
if you had a magic number, what would it be? Or you can write to me and tell me.
I'd be happy to discuss that ad nauseum. There are numbers for like endowing chairs versus starting programs versus long-term research programs. You know, we can, you know, I guess I can give quickly a range before I hand off the talk. You know, with the PIPSEQ stuff that Davey talked about that Carmen did with the LBCRF, that was around $8,000 to get that little platform off the ground. It let me hire Carmen in the first place just to get her in. And then I was able to juggle some other funds. It let us buy the platform. To do some of the larger sequencing based studies to say characterize LCAS, that we're talking hundreds of thousands of dollars. And then if we're starting to talk about maintaining personnel long-term, that's 2/3 of my budget is personnel. And these people are all highly trained. You know, I have 4 PhDs in my lab and they're all still underpaid because of academia. But then we're talking, you know, hundreds of thousands of dollars over multiple years. So I can do a lot with a little. Equipment. So there's cool stuff in the $5,000 to $40,000 range that, you know, has been tough for us to justify from a risk ratio. I think when Joe's on next to talk about metastasis, I am quite confident that Joe could tell you a list of equipment that he would buy with $40,000. I'm looking over him right now. Joe's nodding. There's some cool stuff with like tracking circulating tumor cells that we could do with. I think those machines are in the $25,000 to $40,000 range. That right now we're kind of outsourcing and kicking around. So there's a lot of stuff we can do with varying amounts of support. And I don't know if I actually answered your question.
Noted.
Thank you for asking.
Not at all. We have to support you. Thank you very much.
Thank you. If anyone else has questions, I'm going to hand off for the sake of time, but please, I'm always around. My contact information's in the follow-ups. Please always reach out. But I'll hand it off to our next presenter. Thanks, gang.
Yeah.
All right. I am just going to share the screen real quick. And can everyone see the screen with Joe's poster?
Hi, everyone. Well, thanks for the opportunity to talk to everyone today about some of the animal work we're doing in the lab. Just wanted to start off by saying we don't take any of this work lightly. We view these mice as our collaborators to a degree because they give a lot and we can get a lot of information from them. So everything that we're doing here is supported by the university. It's all regulatorily cleared internally and externally. The university works with federal authorities on making sure that everything we do with these animals is appropriate. So one of the models that we're talking about is mind modeling. And what we do there is we actually inject tumor cells directly into the duct. And you can see from this blue kind of dye that's here is we're actually injecting tumor cells and dye into the entire ductal network. That's unique in this model in that it allows the tumor cells to grow in the tissues that they normally come from. A lot of the time, we will just inject tumor cells directly into the mammary fat pad. And that's good, and it grows in the right location, but the cells that are around it and that they're used to growing around are not very typical. So what we found is that this model and supplementing the mice with estrogen to model postmenopausal disease is actually a better model of tumor growth. What we have in the middle panel here is we can actually measure these tumors on these mice using something called luciferase. Luciferase is an enzyme found in fireflies, and it's what makes them glow at night. So we can inject substrate into them, just goes into their belly. All of those mice are just sleeping. They just have a little bit of anesthesia for the time being. And then they glow like fireflies, and we can put them in a specialized camera that allows us to track their growth over time. The other thing that we can track through that manner is how they disseminate and metastasize throughout the mouse to other organs. You can see that in some of these pictures here. These are what the organs look like after the tumor cells have spread throughout the mouse. There's lung, liver, spleen. We see gynecologic tissues such as uterus and ovary, and GI, all the places that ILC cells like to go. And that's really the goal of these models, is to make a model that we can study. We don't have one except for these models. They're just not present in the field, and that's what we are trying to develop. What we have also moved to is isolating brain and bone. Those are two of the most common places that breast cancer goes to, but they're unique to ILC biology as well. So what we found is that in the bone in particular, it's not like intraductal breast cancer or no special type breast cancer. The tumors that typically form lumps create a lytic disease, and they break down bone, and they can actually create a hole in the bone. That's not what's happening in lobular carcinoma. In lobular carcinoma, there's been a lot of anecdotal information in the clinic, but we're trying to follow it up in these models that shows that those tumors actually make and break down bone in kind of equal ratios. That creates a unique environment in the bone that we need to address to get rid of these tumors. Bone is the most prevalent site of metastasis for lobular patients, and that's why we're interested in tracking it. On the other side is brain. For ductal carcinomas, the tumor cells typically go to the parenchyma of the brain, which is kind of the brain tissue that most people think of. Lobular, on the other hand, will go to those tissues as well, but it also goes to the layers around the bones, or around the brain, I'm sorry. So there's actually a thin layer of fluid-filled tissue around the brain, between the brain and the bone, and that's where lobular tumors like to go. And we're not sure why, but it's a unique site that probably requires a unique intervention for lobular patients. So we've started studying those as well. To try and get to that question, we're not just trying to say, okay, we made a model and our work is done. That's certainly not what we're doing. So in the middle here, RNA-seq analysis is trying to determine what genes are changing and what proteins are changing and what makes these different from ductal carcinomas to lobular carcinomas, or what's just completely de novo different in lobular carcinomas. So there we're trying to identify what genes are different in lobular. Can we target them? How do they interact in the cancer cells, or how do they interact in these unique environments? So that is what these two panels are looking at. And we've identified a few genes that might be interesting. We're still following up on those, and kind of what pathways are used in those cells. What can we look at targeting? How can we change these from being blasted? Is standard of care in bone metastasis biology even applicable to lobular patients? Those are some of the questions that we're starting to identify. And down here is some histology of what these bone metastases in the mice actually look like. MM134 and 44PE are two main workhorse type of tumors. And what you can see in this bottom one here with 44PE, right here, if you can see the cursor, I think. Yes. Okay. So if you can see the cursor, that's actually the very edge of the bone in this mouse. That's the surface of the bone, of the leg bone of the mouse. interacting with the tissue around it. And this purple part in here is actually a tumor. So what that tumor is doing on some of these internal images here are actually growing bone, but on the outside is actually degrading the bone. Well, degrading the bone on the outside of a bone is a bad thing because it's poor for quality of life and fractures and some other things. So that's what we're trying to look for and find out, are these cells still ER positive? Are they still Ki67 positive and some other markers? Here in the MM134, we can actually see this kind of interesting bleb of bone here and some down here in the corner. And that's what we're trying to figure out. What is occurring on those edges that we can learn to look at metastatic tissue differently? So happy to take any questions if anyone has any on the work. Sounds good.
You have a point on fatty acid metabolism
Yes.
in here. Can you tell us more about that?
Yeah, so we're actually just starting to talk about that. That fatty acid metabolism, we know that it goes up. We know that for cells in these stressful and new environments, that they like to use fatty acids for energy as much as anything else. But they can also use fatty acids for a variety of things in the cell. So they can use them to convert them to make nucleotides, for example, which is important in maintaining DNA. They can use those fatty acids for energy. And there might just be something with lobular carcinoma, not just that it comes from breast and that it's a fatty type of a tissue to begin with, but we've noticed that some of the target metastatic sites are also fattier tissues. So omental tissues in the GI and the gut, bone. Some of these bones in some of the aged mice, they change from red bone marrow, which is the bone marrow which makes all the red blood cells, all of the circulating white blood cells. That all changes over to more of an adipose or yellow marrow type tissue as we age. So it could be that the lobular cells in particular like to go to the fat that's within the bone. And then they kind of figure out, oh, I'm in this growth target rich environment, and they stay and they expand. Well, if we can figure out what they're really doing as maybe part of fatty acid biology or something else, general metabolism, that might help us inform how we can look at those cells and treat them.
Can you go get that? Picked out. All right. Should I move this?
Thanks,
one?
everyone.
Thank you guys so much. All right. Next up is Maggie. Hello.
Okay, apologies for that. I'm gonna restart so we're all on the same page. So I was saying that this is just more detail of the presentation that I made earlier. And I'll just kind of walk you through what each piece of this poster is. E-cadherin loss is a hallmark of ILC, and it happens very early in the process from normal healthy breast cells developing into ILC. And so the models that we're using are human mammary epithelial cells, or HMECs. And these are primary cells derived from different donors. And we are looking at how E-cadherin loss changes these normal human breast cells. And so there are I used three different methods of suppressing E-cadherin, and two of them represent what we see in ILC with more of a genetic change in E-cadherin expression. Whereas the third one is just blocking the E-cadherin cell-to-cell communication that I talked about earlier. And so I used all three of these methods, and I did something called RNA-seq, which just looks at the changes in gene expression after E-cadherin is lost. And I found up here, you can see this graph. It just shows this list of pathways or processes or cell programs that are affected by E-cadherin loss. And the top hit being related to cell identity. And so that's the one that I have chased so far. And so far, as I've said, I've found that E-cadherin loss does change the cell identity. And in this figure 5 here, we can see this is a heat map of individual genes, and blue means the genes decrease, yellow means the genes increase. And you can see in this luminal signature across all of the, right here, across all of the genetic loss of E-cadherin models, we see an increase in the luminal gene signature, supporting what we saw when we sorted the cells of that shift to a luminal identity. And the shift was also confirmed by protein expression. This is an experiment called flow cytometry, where you actually can look at proteins at the individual cell level. And so this chart here on the left is the HMEX without any changes, just the normal HMEX. And then on the right here, we have E-cadherin loss in the HMEX. And this the top two quadrants are, if cells are in the top two quadrants, then they're luminal. And if in their bottom quadrant, then they're basal. And you can see that E-cadherin loss shifts all of these cells up towards the luminal side. And so the last thing that I didn't share specifically, but I said that E-cadherin loss also increases cell growth. And one of those things that shows cell growth is this marker CD10. And we can see that CD10 increases in the luminal population, both across all of the cell lines. And down here, when I sorted the cells into luminal and basal, as I described earlier, I also saw that this CD10 marker increased. And so that is just one of the markers that we are looking at. And all of this data and the rest of the data in the publication that I shared earlier are all helping us decide what the next steps are to figure out how exactly is E-cadherin loss changing the cell identity. And once we figured that, once we get a better handle on that, then we'll be able to know something about all of ILC cells, because this is an event that happens very early and is carried, would be carried throughout ILC. So does anyone have any questions?
Yes. So what we're going to do is
Do you know how to pull up the chat?
Yes, I got it. Thanks.
Question.
Yes.
Can you hear me? I was wondering if the E-cadherin loss has anything to do with the formation of the cells. So when I was first reading about lobular, they talked about it growing sheets, but
yes.
the sheets and a lump. So why does that happen? Is that connected to the E-cadherin?
Yes. Yes, that's a great question. And yes, E-cadherin loss is a driver of that single file growth pattern that you see in ILC. Yes, I see it in the cells in a dish when I'm working with them and I knock out E-cadherin, they form all these chains just like you see in histology. So that's a great question.
So why be a lump though and still be lobular?
So that I'm guessing that is probably has maybe some sort of mixed histology, but I don't think it's impossible to, or I think lumps are certainly possible, but is not the typical growth that you see.
Yeah, no, mine's totally been non-typical with HER2 positive and
okay,
morphic, but it's definitely just lobular. They've said it's not ductal.
Okay.
Yeah.
Well, I'll look into that and follow up. I'll be back in 10 minutes. I'll follow up. Okay.
All right.
There we have more. Okay, more people talking about the lump in the chat.
Interesting.
Yeah, that is very interesting.
Well, in the sake of time, we are going to move on to our next presenter, and that is Dr. Taylor Benson. Here is her poster, and here is Dr. Benson. So I can minimize this if that's in your way.
Okay. Hello. So we're going to change gears a little bit. My project is looking at a protein called Wnt4 in ovarian cancer, and I'll try to connect it back to lobular cancer at the end. But Wnt4 is a protein that's necessary for development of the female reproductive system and the breast. And then there are inherited genetic variants in the DNA that encode Wnt4 that are associated with increased risk of gynecologic disease, such as endometriosis and ovarian cancer. So these variants in the Wnt4 gene are inherited differently across populations. They're inherited most frequently in East Asian populations, which have an increased prevalence of both endometriosis and ovarian clear cell carcinoma. And then high levels of Wnt4. So when you have this variant, you have high levels of Wnt4, and high levels of Wnt4 are associated with poor ovarian cancer outcomes. So we're looking at what Wnt4 is doing to be able to address these disparities. So how am I doing this? Cancer cells need fuel to be able to grow. So we're looking at the fuel sources that cancer cells are using. So we're kind of asking, do the cells use sugars and do they use fats? And I've kind of likened this here to a car engine that either takes gas or diesel. And then we're looking at the rate of their growth with either gas or diesel or lipids or sugars. So what is it doing? You might have heard mitochondria is the powerhouse of the cell. The mitochondria is responsible for turning fuel sources into ATP, which allow the cell to proliferate or grow. So we actually find that Wnt4 is localized to mitochondria. And in the presence of Wnt4, the mitochondria are healthy. So I'm not sure if you can see my pointer, but they should form these kinds of networks. And then when you take Wnt4 away, the mitochondria become fragmented and broken. So these Wnt4 deprived mitochondria are no longer able to make ATP or energy from the fuel source that we give them. So we see this when we culture the cells in normal media, so in media that has all of the essential components, so fats and sugars. These cells are able to grow normally. So that here. But when we take away lipids, the cells will actually, the mitochondria will degranulate and the cells will die, or at least continue or not continue to grow. So we're hoping to be able to leverage this to understand cell growth and how lipids are being used, maybe in the absence of glucose. Like in fat-rich tissues like the breast or in the ovary. And yeah, we're hoping to take this back into ILC research because Wnt4 is an important component in that research as well. So let's see. Let's take any questions.
Yeah. Here, let me pull up the chat, but we're happy to take any questions. I also just wanted to point out, I just spoke with Matt, and he said that it is a spectrum for ILC in developing lumps and that there can definitely be solid masses associated in ILC. So I just wanted to let everyone know. All right. I will go grab the next speaker if there are no further questions for Dr. Taylor.
Oh, can I ask a question?
Yeah, of course.
So does this suggest if you've had fibroids, you're more likely to develop ovarian cancer?
I'm not sure if it's more likely, but the presence of Wnt4 makes you more likely to develop all of these things.
And Wnt4 is associated with all of those.
Yeah, not necessarily with each other. Yeah.
Yeah. Thank you.
That's a good question. Yeah.
Hi, everybody. Sorry about that. My name is Maddie. I was a tech in Matt's lab for 6 years. I'm actually now a graduate student here at CU Anschutz. It's all Matt's fault, so you can blame him that he inspired me to go on and do bigger things. So this poster here was actually presented. I don't know if you caught what Davey said, but I had the opportunity last March, I believe it was, to go to the Colorado State Capitol and present this poster for the public, but not more importantly, but in addition to our state representatives. And I actually did have a couple come by and we got to talk and chat, but really just trying to enforce how important the work that we do here is and why they should give us money to do it. So, because otherwise, unfortunately, we don't get to do that. But, and yeah, so it's very, very basic, very simple. I know that you guys have gotten a lot of other posters today from other people that kind of go more in depth on these two topics. So the two projects that I was primarily working on was the DNA damage repair pathway that Joe did, and then also went for. So I know Taylor just talked a little bit about that in the ovarian context. So yeah, I don't know if there's a preference. I can kind of walk through the posters very briefly, or I can just take questions on it. I don't know what the general consensus is on that. If anybody wants to speak up, I'm fine doing whatever. I know I came in late, so if we just want to be like quick with it, that's fine too. But I guess if everybody's cool, I'll just kind of quickly go through it. Does that work? Okay, great. So the two projects that I was primarily working on were the DNA damage response and repair pathways and went for. So just very quick, we'll go over the DNA damage repair mechanism. So I'm sure you've heard, but in ILC, those cells like to repair their DNA in a different mechanism than IDC. So that's called non-homologous end joining is the way that they prefer. And I like to call it the duct tape and spit method. So normally if you think about like your cells as a car and you get damaged. So your DNA has been damaged, your car is now damaged. For the normal or the IDC way is they would take your car to the mechanic, they would fix it up all nice and new. It's exactly how it is when you brought it in. That's called homologous recombination. What ILC prefers to do is, so your car is sitting on the side of the road, it's smoking, it's damaged. You go in the back of your trunk, you grab whatever random toolkit you have, you kind of tape it together, and then you keep on driving. So that's homologous, non-homologous end joining. And that is what ILC prefers to do. So we're not really entirely sure why ILC prefers one method over the other. And so that's kind of what we've been trying to figure out. In addition to how can we exploit this in the sense that can we use targeted therapies with this method of DNA damage to further kill the cell? So what Joe's been working on a lot of, and that I kind of started working on in the lab very early stages, was looking at talazoparib. So that is a PARP inhibitor, which is a DNA damage repair drug. So we found that when your DNA is damaged, we were able to use talazoparib to kind of come in. And essentially what it does is it's taking away your trunk toolkit. So you can't really actually repair your car and it's still kind of messed up. Really interestingly here, what we saw, especially early on, is that when treated with talazoparib, ILC cells become more sensitized. So when we had IDC treated with talazoparib, they might have been a little bit like stalled in their growth, but they were able to overcome that assault on their DNA damage and keep growing totally fine. ILC was the opposite. So they were treated with talazoparib. They kind of stalled and they like kept being stalled. So they didn't die, but they didn't grow either. And that's where we kind of got this idea of, well, this drug is doing something. What is happening? And can we use this as a potentially effective treatment? Which is then where Joe kind of took it and he's been using it in mice. And then that's where they're trying to get with the clinical trial with talazoparib. So that's where that currently, or for me, when I left, that's where that currently was. Any questions on that, or do we want to wait for questions until the end? Okay.
Can I ask if that's kind of the first project?
Yeah.
It's a good time to ask. Is it, to what degree is, would that apply, or is that another research project to HER2 positive? Because I know mostly it's ER positive. So does this, because I've been told, we're just treating you as a HER2 positive
Mm-hmm.
rather than thinking about the lobular side of things. But I'm always wondering, well, could it be relevant that it's also lobular? And what you just described about the difference suggests that it would be.
Yeah, no, that's actually a really good question. I am not positive on that because, like I said, I've only had the opportunity to work on this specifically in our ILC cells, which are classified as HER2 negative, and the cells that we do work on are HER2 negative. However,
we do have one cell
Natalie
line.
took this over.
It's my current rotation project. We do have one cell line that is HER2 positive, so it is kind of that outlier of ILC. We haven't really tested them in this context because we're really just trying to, kickstart that clinical trial and show that it will work in traditional ILC models. But it would be something really interesting to look into about how that extra HER2 receptor plays into the biology of all this. And if that, is a compensatory mechanism for the cells, or if they will still be sensitive to the treatment.
Yeah. Were those the 1428s?
Yes.
So yeah, I did do a little bit of work in the 1428s.
Those are bracket 2 hypomorph.
Yes.
It's a different line.
Okay. I haven't worked on that cell line. That one came in after I left. But yeah, as you can see, the work is still being done. And the question exactly is there, and we are trying to answer it. If you haven't, figured it out yet, Matt likes to do a little bit of everything. And if we could clone him, we would love to. So there were just, 50 of him, and we would have even more projects to do. But yeah, so that, like I said, that's kind of where I stopped. And obviously it's really cool that this drug has now been taken to mice. They're trying to work on getting the clinical trial going for it. And Joe has seen some really, really, really just fascinating and encouraging results with the drug in mice. So then the second project that I was working on, which is very near and dear to my heart, is WNT4. So WNT4 is a protein that is crucial for the development of female tissues, such as the breast and ovaries. And I know Taylor talked a little bit about this before, so I won't go into too much detail for what WNT4 is, but what the work I've worked on with WNT4 is kind of the before and how we got to what Taylor's project was and what she's working on right now. So typically what WNT4 does is it is made in the nucleus, it's transferred out of the cell, and then it will bind to these receptors that are on top of the cell that then indicates all of this downstream, cellular, go do your thing. We saw in WNT4 specifically for ILC that when you try and stop that mechanism that gets WNT4 out of the cell to then go and cause these downstream effects, you would think that, okay, well, that would mean that the stuff that WNT4 activates is no longer activated. That is not the case in ILC. So we kind of explored that a little bit further. Davey did a lot of work in this initially, and she's actually the one who found that if you try and stop the mechanism, WNT4 is still active. So what we were looking at is, well, we know it's active. Where is it going? What is it doing? And we saw that WNT4 is actually able to move within the cell and is able to activate pathways that it would otherwise be unable to if it behaved in the way it's supposed to behave. So this is an atypical pathway. We saw that, or had an inkling that WNT4 was actually interacting with the mitochondria, and that's that connection to metabolism that Taylor talked about. But we were trying to kind of figure out, well, how do we see this? What do we do? How do we know? And we use this method called BioID. So it's essentially a GPS for your cells. And I don't know if you've seen those videos where you have a cat and they stick the little GPS tracker on them and they can see where they go all around their neighborhood. But that's basically what BioID is. So you have your cat, which is your WNT4 protein. You stick on a little GPS collar, and then which is biotin, and then you let it go. So you let the cells go and you see where your cat, or WNT4, goes throughout the neighborhood. So where is it going within the cell? You're actually able to see which proteins it interacts with, which proteins and other things it doesn't interact with, but also within that interaction, or the proteins that it interacts with, you can actually see which areas it interacted with the most. So that you got those highest hits. And from this screen, we actually determined that WNT4 was really localizing or hanging out near the mitochondria. That was our strongest hit, which was just mind blowing. It should not be anywhere near the mitochondria. And that is kind of what took us down this road where we're like, okay, so mitochondria Wnt4, was very likely that Wnt4 is involved with metabolism of ILC cells or in ILC cells. So kind of trying to further explore that idea of, we know that ILC doesn't prefer glucose, right? That's why FDG PET is difficult, or normal scans are difficult, right? Because you have that radioactive tag glucose. ILC tumor cells don't really take up glucose. So this kind of starts to poke at that question of, well, what is the cell doing? What does it prefer to eat? And I'm sure, as Taylor mentioned, we saw that there was a similar, or Wnt4 behaved similarly in this Wnt4 variant in ovarian cancer. And we have some better established models, both cell lines and mouse models in ovarian, that we were able to then take this idea of we need to explore this metabolism idea further. And then that's where Taylor's taken it into the ovarian cells with the hope that whatever she finds metabolism wise or whatever she does find, we can actually bring back into the ILC context and try and apply and see, is it the same, which it's very likely, again, very similar. But yeah, understanding this metabolic kind of pathway or interaction with Wnt4 can help us then further both understand the biology of Wnt4 and how we can create better targeted treatment therapies for it. But yeah, so that's just the very, very basic of my poster. Like I said, it was very, I tried to make it as simple as possible because I was presenting it to our representatives who many don't have a science background. They're all laws and bills and everything, and I don't know anything about that. So, but yeah, it was a wonderful opportunity. And yeah, does anybody have any questions?
Okay. Hi, everyone. I am Carmen. I may have met some of you last year, either in person or virtually. If you were at this event last year, you've already seen this data. I presented this exact same poster last year. So this particular data is a bit old, but I still think it's important to highlight the importance of donor support and what that has done for the Sikora lab. And that's showcased in my work. So directly because of donor support, Matt was able to hire me and we were able to purchase the equipment necessary to do something called single cell RNA sequencing within the Sikora lab. So to kind of go into single cell RNA sequencing and what's so cool about it, in a lot of the things that we do to investigate ILC or any kind of cancer, we're looking at the cancer as a whole or looking at a given tumor as a whole. But just like no two cancers are going to be the exact same, you also get heterogeneity within a given tumor or a given sample. So using single cell RNA sequencing, we're able to tease apart a greater sample to identify different populations within that sample. So using this technology, we look at each individual cell. We kind of create a profile for that cell based on all the genes that cell is expressing. And then we're able to compare cells to one another. In doing that, we can identify new states of ILC that we otherwise wouldn't have seen using other technologies. And by focusing on those, we can learn more about ILC in general, but also look at new ways of targeting ILC by looking at specific populations. So I'm happy to answer any questions about single cell RNA sequencing, about ERMDC1, about DNA damage repair. I'm not sure what the conversation with Maddie was, but I'm the person for that. So does anyone have questions? Just get the chat.
Yeah, I know, right? Oh, let's see that.
I can't remember who asked the question.
I think it was Pam.
I was asking if you could just give us a high level summary of what this newest class of anti-estrogens are, and I think yours was like how it affects the lobular cells.
Yeah, so I looked at 3 new
SERMs and SERDs. So elacestrant, lasofoxifene, and camizestrant. And I was comparing them to the two current standard of care, so tamoxifen and fulvestrant. And just overall, looking at the results, we found that camizestrant looked very similar to fulvestrant, which was super exciting because fulvestrant works great in theory, but it has very low bioavailability, which makes it not a great solution for all the patients. Also, like some people just have terrible side effects with it. But with Cami, it's a lot more bioavailable. And from what we've seen, it does a really similar job regulating those estrogen receptor regulated genes. And then we also saw that lasofoxifene had pretty similar activity to tamoxifen. And I believe the other, both of those, lasofoxifene and camizestrant, are still undergoing clinical trials. But the third one that I looked at, elacestrant, which had kind of mixed results, that one has been approved. Yeah. Is there any more specific thing that I could talk about with that?
What's the mechanism of the elastrin? Is it similar? Is it similar to what the AIs do, or tamoxifen, or how does it?
Yeah, so it's pretty similar to tamoxifen, where like in a traditional cell where you have the estrogen receptor, when estrogen binds to the estrogen receptor, it kind of gives the cancer cell a big thumbs up. So what we do is we send in these treatments of like SERMs and SERDs. And so like what elacestrant will do is instead of estrogen binding to the estrogen receptor, it will bind instead. And by binding instead, it will modulate that activity and regulate those genes. But one thing we've seen is that specifically with tamoxifen, it might not be as effective for ILC patients. So that was something that I was kind of looking at. I was comparing ILC and IDC lines to kind of see if there was a big difference. And we didn't pick apart anything major from the work I did this summer, but I think that's something that the lab is still looking into.
And the way it presents itself as in like thread-like, it's still able to adhere to the receptors?
Yeah, it still binds to the receptors the same way in ILC and IDC.
Thank you.
Yeah.
Perfect timing. He just got free.
Hi. Thanks all for hanging on so long. Are there any last questions I can ask before we sign off officially online?
I might just ask if you could say a little bit about ctDNA testing and explain how that's used or helpful in any of the research or understanding, particularly metastases, and how it's supposedly the cells just circulate around in the bloodstream and you never know when they'll appear, is how I hear it. And I think, well, if there's some way to do a liquid biopsy, would that be somewhat reassuring that there's not that much circulating? I don't know. Just trying to understand that.
Yeah, so there are two different circulating markers that you alluded to in there. There's circulating tumor DNA, or ctDNA, and circulating tumor cells, or CTCs. The CTCs are exactly that. They are tumor cells that have probably broken free from the primary tumor or metastatic site and are floating in circulation as a viable tumor cell. Those patients with ILC tend to have a higher CTC burden, is what it's called, or the number of cells in circulation is higher, and it's substantially higher than ductal cancer patients. But we don't know if that's a bad thing yet. We know it's higher, but we don't know if that means that it's better or worse, or if it's just a feature of the fact that the ILC cells can survive in circulation differently. ctDNA may or may not be from a viable cell. It can also be from tumor cells that have died and just shed their DNA into circulation as part of their death. And that's because that's routine. We can do cell-free DNA testing for even maternal and pregnancy-related things. So having DNA in your blood's not a bad thing. So how we use either of those things clinically in theory, though, is that you can track the presence or absence of those or the change in relative levels as whether you have a response to therapy or eventually a recurrence. So for ctDNA, for instance, we know that before therapy, during therapy, and on therapy, you can actually see a decrease in ctDNA, or you see an increase first as cells die, and then it tapers off and hopefully goes down very low. And then hopefully it stays low. And if it ever comes up, there may be a recurrence. And it's not dissimilar for CTCs. They're just two different ways to capture tumor features. The nice thing with ctDNA in theory is that you're not just trying to scan one particular organ by, say, PET scanning or something like that. It should represent any metastasis throughout the entire body. So you should be able to use ctDNA or CTCs to essentially surveil the entire body. I mean, the catch is we don't know where it came from if it lights up. But it's a better way to capture potentially the entire body. We know from a number of studies that if ctDNA and CTCs were low and then come up, it does precede by months to years the clinical presentation of a recurrence or a metastasis. So we know it means something, but where the field is at right now is that we don't know how to best act on that information. The question is, let's say you're on fulvestrant for like a second line endocrine therapy for ILC. If you're on fulvestrant and detect an increase in ctDNA or CTCs, if you might say, okay, my tumor is resisting, I need to switch. But are you actually going to get more benefit from the next line of therapy, or are you just shortening the duration of benefit from the first therapy that you're currently on? And that's where the trials are stuck right now. But they're actually asking that question. So there's some really cool trial designs that were announced at the San Antonio meeting last year, where they're doing that sort of switching and asking not only overall time to progression, but how much benefit are you then getting from the second line of therapy? Are you still getting months to years of benefit on, say, a CDK4/6 inhibitor or whatever you would switch to in that setting? So hopefully I answered your question, but in general, we're trying to use it to track it. And you can use different features. And then hopefully in the next few years, we'll know how to best act on those changes. The catch will be for Lobular is that with those higher burdens, we won't have data on Lobular specific settings for a little while. So there are several groups that are trying to get that data.
Thank you. Very helpful.
You're welcome. Hey, Helene, I think you have your...
Yeah, I'm just wondering, and it's probably linked a little bit to the sort of circulating tumor cells, dormancy, is there research being done on that, and what people can do to prolong dormancy, I guess, and equally keep their circulating tumor cell levels low? Is there any work being done on that?
The short version is yes. There are a couple European groups actually. Val Brunton in Edinburgh is actually particularly active in ILC microenvironment and dormancy. So I don't have great answers for you for how Lobular behaves in that sense yet, but we do think dormancy is really important for why cells can recur late. My own models in my head right now are that lobular cells are ridiculously endocrine dependent, but they're also really good at just hanging around without estrogen present until they see it again. So my hunch is that when we eventually have data from the post 5-year endocrine therapy trials, like Atlas that extended endocrine therapy past 5 years, my hunch is that Lobular will show a greater benefit of extended endocrine therapy, but I don't know that by any stretch yet. But my hunch is that the Lobulars, for some reason, are just really good at hanging around in the presence of endocrine therapy. And then as soon as estrogen comes back, they can wake back up, which is one of the reasons that we're looking at things like PARP inhibitors and things that will kill the cells and not just let them wait around. But I'm hand waving. I don't have a good clinical grade answer for you yet.
Thank you.
Anybody else before we sign off? And you're all welcome to contact me after the fact. I'm happy to chat anytime really about what we're doing, about our research and how we can support you and especially if you're interested in getting integrated into Front Range ILC.
Thank you very much.
Thank you for joining us. I'm going to glance at Natalie. Are we ready to sign off then? Okay. Yes. Thank you all for sticking around. yeah, we really appreciate you joining us.
Thank you.
Bye-bye.